Hello everyone, my name is Vladimir Nikolic and I will be presenting GoldRush-Edit, which is a targeted, alignment-free polishing and finishing pipeline for long-read assembly using long-read K-MERS, which is a long title. So long reads are very widely used in genomic applications, but they still come at appreciable error rates. So they would include mismatches and indels. And this is an issue if you want to do, say, genome assembly. Now there are polishing methods out there which would fix these errors, such as rack-on, but depending on the genome you're working with, they might require a lot of memories. They're not necessarily scalable, and they require large memory servers. And so here we present GoldRush-Edit, which is a memory-efficient polishing pipeline to correct these errors in long-read assemblies using a scalable and targeted K-MERS-based method. So the way GoldRush-Edit works is we start with input sequences, say contigs from genome assembly, and we map long reads to these contigs using, by default, anti-link, but you can substitute another mapper in there. And then we split these mapped long reads into K-MERS. Now we make a Bloom filter for each of these contigs and insert the corresponding K-MERS into their Bloom filters, so that each Bloom filter for each contig has the K-MERS from the corresponding mapped long reads. Then we use these Bloom filters with anti-edit, which is a fast polishing tool to fix mismatches and small indels, and for the regions that it cannot fix, it simply marks them. Then it passes the sequences to Sealer, which is a gap filling tool, but it treats these unresolved regions as gaps and then tries to fill in the gaps. Now, both of these are individual tools and they can be run independently. Now, to maximize performance, we have a dedicated pipeline spawner process which launches pipelines of anti-edit and Sealer, and it launches them up to a limit so that we don't use too many threads depending on what the user wants. And then we have a dedicated Bloom filter building process which builds Bloom filters in the background and passes them to these pipelines. And finally, we have this Reaper process that collects the results and outputs them to a file. So we have this multi-process environment with well-coordinated processes working together. So for results, we have on the left side, we have the polishing quality plots, and on the right side, we have resource usage plots. So for the polishing quality, on the y-axis, we have indels per 100 kilobase pairs, and on the x-axis, we have mismatches per 100 kilobase pairs. And so we start with baseline, which is the gray circle, which is the genome assembly that is still unpolished. And then the blue circle is that assembly polished with GoldRush-Edit using anti-link mappings. So we have a decent improvement in quality. But further, if we use something like Minimap 2, which is perhaps more accurate, we get further improvement in polishing quality. And in the red circle, we have polishing performance of rack-on. So depending on the data set, we can get pretty close to rack-on. These data sets differ. These are 3 human data sets, but they differ in their sequence quality. So NA24385 has actually the best quality, and so we have more opportunity to fix errors there. However, on the right side, we have a very drastic difference in memory usage between GoldRush-Edit and rack-on. So rack-on for all 3 datasets uses about 600 gigabytes of memory, whereas GoldRush-Edit, regardless of whether you use anti-link or Minimap, uses up to 30 gigabytes or so. So for Minimap, it might use 30 gigabytes, and for anti-link, it might use about 15 or so, where using Minimap uses slightly more time to finish. So in conclusion, GoldRush-Edit is capable of outputting draft assembly quality comparable to its competitor rack-on, but at an order of magnitude less memory usage and comparable runtime. And it's possible to use alignments for polishing, so from Minimap, let's say, at slightly increased runtime, but also improved polishing quality. So I'd like to thank the funders of this project, Genome Canada, Genome British Columbia, and National Institutes of Health. You can find this project at GitHub at this URL here, or you can visit our lab website at this URL, or you can contact me at my email if you have any questions. Thank you.